check whether its link is correctly streamed or not.
Hasn't started yet, but
Okay.
reload.
Okay.
It should start.
If you're an adult in Alberta and you need a
That was the ad before it starts, so I think it's working.
Yes, it's working. Okay, so we are live now. Soma, hit the recording option and record in your own computer, not cloud.
All right. Live and recording.
Okay. Shoma, just before starting Liberty, for the Liberty, you pronounce our speaker name. It is C Bansal. Okay, Liberty or Shoma?
Yeah.
I just wanted to make sure because I have to say your name so many times in the introduction.
Yes,
so
Bansal.
It would be embarrassing.
No.
Yeah, you can get it from the person also. He's sitting right here.
I know, but I feel rude asking him how to say his own name.
No, it's all right.
You've already said it many times.
Yes.
Okay.
Bansal is fine.
Bansal.
Okay, so we should start. Soma, change the view to speaker.
Yeah. There.
Okay. Thank you everyone for coming and joining our 28th webinar in this series. We were very fortunate and very happy to have Dr. C Bansal sir as a speaker for today's webinar. I request Liberty to introduce Bioingene and our speaker, then Jacinta will go for question answer round. We are collecting the questions from the YouTube live chat box, and we already collected few questions on GM crops and its regulations and everything from our initiative or campaigning through our website. So we are going to ask our speaker after his presentation, and Soma will do the interview section after the presentation and questions round. And I will give the password for today's certificate collection during the interview session. So keep patience. We already sent the link for the certificate collections. If you don't have it, you email back me. I will send it again if you missed it. And I will send once again within 10 minutes to collect for the certificate collections. So I request Jacinta, for Liberty, introduce Bioingene and our speaker, Dr. C Bansal sir.
No problem. Thank you, Shivo. Excuse me. Welcome everyone to Bioingene webinar series. The webinar series is a platform from where plant scientists can present their research to the world and future scientists can gain knowledge, perspective, and inspiration. The appreciations that we receive from our global viewers after every webinar keep us motivated and have led to the constant growth of Bioingene with new viewers and new endeavors. We are very excited to announce our new endeavor, the Bioingene Plant Sciences Journal. This Bioingene Plant Sciences Journal is an online, international, open access, peer-reviewed scientific journal. It is a monthly publication and completely online. We seek to publish papers that are relevant in the fields of plant science research and address the present and near future scenarios of crop health, yield and production, and different biotic and abiotic challenges. Aspects such as food security, nutritional security, effects of climate change, crop resilience, application of artificial intelligence in agriculture, genome sequencing, genome editing, omics, technologies, CRISPR-Cas, modern breeding techniques in plant sciences will be highlighted in this journal. Selection of manuscripts for publication will be based on novel research, original data, synthesis, and concepts without plagiarism and violation of any copyright. We welcome submissions from all fields of plant sciences, including agriculture, and from any source. Please visit our Bioingene website to know more. Now, coming back to today's event. Today is our 28th webinar of the series. We welcome all the viewers who have joined us from around the world. After attending today's talk, you can apply for a certificate of participation via the feedback link and password. We have already emailed the feedback certificate application link to the registered participants. The password will be provided multiple times in the YouTube chat box during the question and answer and interview session. Submission of the feedback form is mandatory to receive a certificate. When you fill out the feedback form, please mention the full name of your institute and the full institute address. You can collect your participation certificates after 2 days from our website. Bioingene does not send certificates through email. Please visit our website to register for upcoming webinars. You can connect with Bioingene via Facebook, YouTube, Twitter, and LinkedIn. Excuse me. The topic for today's webinar is Germplasm to Genome Engineering, My Quest Towards Achieving Big by Professor C Bansal. Professor Bansal obtained his Ph.D. with gold medal from India's premier institute, the Indian Agriculture Research Institute in New Delhi. After obtaining his Ph.D. degree in 1989, he pursued advanced research from Harvard University in Cambridge in the United States. Since his return from the US, he occupied eminent positions in ICAR, in the Department of Agricultural Education and Research by the Government of India, of Professor of Plant Biotechnology from 2004 to 2010 at IARI, New Delhi, and was a coordinator for national projects on transgenics in crops from 2006 to 2010. He was also the director of the National Bureau of Plant Genetics Research, National Bureau of Plant Genetic Resources in New Delhi from 2010 to 2016. Currently, Professor Bansal is Secretary of the National Academy of Agricultural Sciences and a member board of directors on the Global Plant Council. He was the first to become selected for the prestigious Norman Borlaug Chair for Crop Improvement by ICAR. He has contributed significantly in the area of plant biotechnology, has published over 150 research papers and book chapters. Professor Bansal's research findings and outcomes are cited and used in crop improvement programs by several laboratories and institutions globally. Professor Bansal has won several awards at national and international levels. To name a few, first, the highest award of the ICAR for an individual's contribution, the RAK Award. Secondly, an award of the Indian Science Congress conferred by the Honorable Prime Minister of India. The third is a Biotech Career Award given by the Rockefeller Foundation in the United States. He also received a recognition award of the National Academy of Agricultural Sciences and the prestigious Haryana Vigyan Ratna conferred upon by Honorable Government and Chief Minister of Haryana. In February 2019, Professor Bansal was awarded the second highest award of the National Academy of Agricultural Sciences by the Honorable Minister of Agriculture in the Government of India. Professor Bansal is a recognized popular science teacher and research guide. He has supervised over 20 doctoral students. Many of his students got national level awards, such as the Best Thesis Award of the ICAR and the National Young Scientist Award and the IARI Gold Medal. As director of the NBPGR, Professor Bansal was conferred upon the Research Leadership Award for his outstanding contributions in research, education, coordination, and management. We are highly honored to have Professor Bansal with us today, and it is my request to all of our viewers to please enjoy the opportunity to listen to him speak. I now request that Professor Bansal to share his screen and begin his talk whenever he is ready. Thank you.
Yes, sir. We cannot hear you, but we can see your screen.
Oh, your mic is on mute.
You ask, okay.
Yes.
Now.
Good. Now it's okay.
I'm trying to figure out my slides, whether they're moving ahead or not, but it's not moving ahead because automatically it came on the slide mode, which it should not have.
You just
so
share
you can
your
use
desktop,
yeah, you can use your keyboard, sir, for
Yeah, that's
left
what
and
I'm
right
doing.
key.
No, that's what I'm doing.
Okay.
1 second. Shall I try again? If you.
yes, yes.
Yes, sir. No problem. Take your time. No
Yeah,
problem.
it's all right.
Yeah.
Just when the screen sharing start, just create the share the desktop.
No, 1 second. 1 second, because I think I should go back here.
Choose enter screen option, sir. When you try to start the screen sharing, choose the option enter screen. Okay, now it's coming.
Okay, now
Yeah, yeah.
Yes. Is it working?
Yes, it is there now.
yes.
Yes. Yes.
Well,
Good.
thank you, the Bioingene team, for inviting me to share my experiences of scientific life, in fact. And I'm very thankful to all the viewers and the participants who have registered to participate or otherwise are there to support me as a speaker. And I'm here through my presentation to inspire particularly the young scientists and scholars and students. Bioingene has been doing an excellent job. I convey my congratulations to them. This is the first in the series of this new year, 2021. So I also convey my greetings to all the viewers and the Bioingene team to convey the very best wishes for this 2021 is a new happy year, free of any kind of infection which we had witnessed, unfortunately, in 2020. So wishing you all the best, not only for this new year, for the whole decade ahead. And we all together try to make a difference to make this planet more livable, more environmental friendly, more clean, and more sustainable. So with that view in mind, the title of my presentation, as also it's been advised and suggested by the team in Bioingene, is Germplasm to Genome Engineering. This is the area with which I have been associated in my last 3 to 4 decades of research. And I have been, in fact, trying, not that I have achieved something big, but I must tell the audience and the viewers that my quest has always been achieving something big at whatever level I could. So what is that my concept of achieving big? I thought I would like to share with you the concept of achieving big. The concept of achieving big is this, that we must dream to achieve something big. And while you dream alone will not be enough, you need to have that kind of a passion or also a mission to achieve that dream. And then we must always keep ourselves up to date with the knowledge, with the changing science. It's very important. A combination of all this I have practiced myself have to some extent helped me achieving big, whatever little I could actually achieve. And these 4 arrows are important to always keep networking. Networking with your own faculties, in your own mind and body and soul, and also networking nationally, internationally with our scientific fraternity. So as the title was Germplasm to Genome Engineering, and genome engineering particularly for sustainable development of agriculture, for crop improvement. So I thought I must like to share with you the historical perspective of genetic improvement of crops. You know, it's not only the technology alone. I would like to send this message very clearly across. Important is that we should have a holistic viewpoint of where are we and what kind of research are we doing and where do we fit in, really speaking, in the whole gamut of research which is being carried out world over. So if I go into the historical perspective of crop improvement, particularly the genetic improvement of crops, you see, we have a lot of biodiversity around us on this planet Earth, you know, which includes all, basically, including our own human resources, the kind of biodiversity, you know. We were actually, you know, 5 to 7 species, you know, not only Homo sapiens, but not only sapiens, but many other, but we are only right now, of course, narrowed down to only one species, that is Homo sapiens. But we have a lot of biodiversity when we talk of other, you know, organisms, like you talk of plants, you talk of trees, you talk of organisms, you talk of birds, you talk of, you know, all different organisms. We have a lot of biodiversity. Okay. Then particularly when we talk of agriculture, we have a lot of biodiversity in agricultural crops themselves. The crops could be many, you know, they could be like 30,000 crop species have been identified, which are edible in nature, you know. So that a lot of agrobiodiversity we have around us in, you know, in this world. Then specifically count down to crops alone, you know, we have generally call them as plant genetic resources, you know, which is actually encompassing all that crop diversity we're talking of, be it wild species of a particular crop or all the crops, or land races, the wild species when they were domesticated by human beings, you know, thousands of years ago, you know, because agriculture started somewhere 10 to 1,000 years ago. So the land races, you know, are there into this gamut of plant genetic resources. Then farmers' varieties, they themselves started selecting the best, you know, whatever was available that time, you know, and they were growing it for years and years. And then sometimes in 1900, as you all know, the laws of genetics, you know, came into picture, Mendel's laws, and that gave us the science of modern plant breeding. And so we were able to develop modern high yielding varieties, okay? And this was all done through crop crossbreeding, or as we call it as conventional breeding. So this is all very important as we have come down from like say 10 to 12,000 years to these conventional breeding tools based on principles of genetics. And that actually gave us in 60s, I'm talking of India or Asia or Southeast Asia, particularly. See here, that gave us green revolution with the efforts of Dr. Norman Borlaug, you know, at CIMMYT. And that's this green revolution, particularly, you know, with wheat, again, gave us high yielding varieties, also in rice. And actually, you know, this gave us a production, you can see from here, 1950, you know, which was only about 50 million ton. It went up to, in 2010, let's say about 60 years, you know, more than 250 million ton. And today we are able to harvest in India, I'm talking about 295 million ton in 2020. So this in 60 years, you know, we have traveled a long way. And conventional breeding methods combined with the use of these plant genetic resources has given us this particular, you know, potential production close to 300 million tons. And India is in a very happy situation with regard to food security, okay? But then what has happened after all this? After all this, in 60 years, like 60 to 70 years, if you compare from 1950, what has happened? There's a loss of crop diversity. Loss of crop diversity, I'm talking about in farmers' fields. You know, the farmer who was growing hundreds to thousands of cultivars, for example, thousands, I'm talking in case of rice, sometimes in 1900 to 1960 or 70 or before Green Revolution, has come down to only 5 to 50 cultivars in the last about now say 40 to 50 years after the Green Revolution, because the farmers would prefer these high yielding varieties. And this 295 million ton predominantly includes mostly wheat and rice, and of course pulses and few core cereals and others. So we have witnessed a loss of crop diversity to a great extent. And then also we have witnessed an adverse impact on environment in terms of depleting water table, which has gone further down into the underground portion. And also we have come across the problem of alkalinity. Why this happened? Because these high yielding varieties were particularly bred for being responsive to fertilizers or to irrigation. So the farmers were naturally trying to produce more and more, were able to, you know, irrigate using underground water, a lot of it. And also we were able to provide a lot of fertilizer input. So naturally it has also caused to some extent problem of alkalinity. And of course, malnutrition Because world over, only 3 crops, wheat, rice, and maize, provide you more than 50% of the total calorie requirement. So only these 3 to 4 to 5 to 10 crops at the most were available to meet our calorie requirement, and which were not really, per se, nutritionally enhanced crops as such. While this is important, remains important, for food security, but malnutrition is also an issue which is importantly to be addressed. So currently, therefore, we have in last, let's say 70 years, if we talk from 1950, we started looking for increasing our production. What in the meantime, in the 60 to 70 years, the climate had changed a lot. We have witnessed it. I have myself witnessed, for example, when I was a student in 1985, how many years ago? About 35 years ago, the carbon dioxide concentration was 360 ppm. Today it is 410 ppm. 50 ppm it has increased in the last 35 years, more than 1 ppm per year. And of course, there's a high temperature, all we know it globally. They're shrinking resources, we're talking of shrinking resources in terms of land and water, Then there's an emerging pressure of many of these abiotic and biotic stress factors, and of course, malnutrition continues, to be an issue which need to be addressed. So what is that, we're trying to do today is actually, address the goal. Today, the goal remains, major goals remain, address the challenges as mentioned here, increase crop diversity, because there's a loss of crop diversity we have witnessed in the last 50 to 70 years. And how do we increase that crop diversity? By using modern science-based tools and technologies, okay? And how do we also increase crop diversity or broaden the genetic base of some of our crops which have become totally narrow because of the reuse of some of those, modern varieties and genetic stocks and breeding programs? We need to bring back the genes which were lost. I'll just show you in the next slide what do I mean by this, and therefore to bring back these lost genes, we must search them back into our germplasm resources. And of course, we are happy in a way because green revolution gave us, self-sufficiency, particularly, this part of the world, I'm talking in India or Pakistan, Bangladesh, some of these countries, and in 2020, December, CIMMIT and other institutions celebrated 50 years of the Nobel Peace Prize awarded to, great scientists, the world has produced in terms of agriculture, Dr. Norman Borlaug. So what do we mean by now bringing back those genes from wild species into our modern cultivars using the tools of modern sciences? So here in this slide, I'm trying to show you the role of plant genetic resources in modern variety development. Actually speaking, these are the wild species shown here, with so many different genes shown in color, like red, blue, or yellow, and early domesticates, which I said, the selection by the farmers themselves, we left behind many of these genes, particularly as shown in yellow, you don't find them here, and then these were further bred based on the modern tools of science, I said in 1900 onwards, based on the principles of genetics and conventional plant breeding tools. Scientists develop modern varieties. I give an example of high-yielding varieties developed for the purpose of green revolution here, as I mentioned. And you can see here again, not only yellow, even the red genes have disappeared. So we have, we have lost some of the genes in our modern cultivars. They are not there. And we need to now bring them back using modern tools. That's what and then do we have these genetic resources conserved? And the good thing is that, yes, we have some of these genetic resources conserved in some of the gene bank. This is just a picture of Indian National Gene Bank shown here. And these resources will act as sources of genes for improving agricultural crops. Okay, just giving an example from Tanksley paper in 1977, a classical paper which appeared in Science, that one of these wild species of tomato, is totally, you can see, green in color. Even when it ripens, it remains green in color. But from this variety, there's a gene which was hidden, as shown here in the wild species, which was then taken and introgressed into this cultivated tomato variety, and it increased its lycopene content. So not necessarily that only the genes, the phenotype which it gives you, the genes are expressing. No, the genes which do not even express at all, they're dormant here in a way. If they can be introgressed into our cultivated genotypes, as you can see has been done in tomato from this wild species, and you can see increased content in lycopene, which is important, which is a very good antioxidant. So therefore, these lost genes and alleles can be recovered only by going back to these wild accessions or what are stored in the gene bank. I'll tell you a lot about it again as I go further. And these then need to be brought back from these wild ancestors to our cultivated crop species. So important is what? Important is that you have these genetic resources, as the basic building blocks, for developing your newer varieties. And what in turn you use is a technology. So both are therefore important, and what are those tools available today to bring those genes back from this diversity of plant genetic resources of wild species we have. Of course, we continue always using conventional breeding, which will remain ever important, in times to come. We have, fortunately, as the climate has changed, we have improved a lot in terms of understanding of the genetics of some of these yield contributing traits or the crop themselves, through genomics approaches and others. And we have been able to develop methodologies of molecular breeding, genomics breeding, then genetic engineering to give rise to GM crops. And today we are talking of world over genome editing, through CRISPR-Cas system, not only of improving crop plants, also in general, the life, of animals and human beings as well. Okay, so, but then ultimately to use these tools and technologies to bring back those genes, you always need to depend on, the germplasm as a source of that genetic diversity and genes, which is important. And so what I'm going to actually travel through my own journey, from germplasm, I highlighted importance of little bit already, to genes, and from genes to GM crops, development of GM crops, and then back to germplasm. Actually, this is my own journey, last 30 to 40 years, which I'm going to travel through with you today. And at this time, I acknowledge, that I got all my training from these two great souls who are unfortunately no more. Professor S.K. Sinha, my professor in India, with whom I did my Ph.D. at Indian Agricultural Research Institute, and Professor Lawrence Bogored, with whom I did my postdoc at Harvard University in USA. So as a plant physiologist, I could, start with germplasm, and then with my training molecular biology, I could do, a lot of things around genes, finally use those genes in developing GM crops. But then again, my passion was to come back to germplasm, and which I did. So I'm going to talk to you one by one with all this. So to start with, actually, I started working for my PhD by importing some of these, Triticum mistivum and related species from Canada, for my PhD, because I wanted to work for drought resistance in some of these species of wheat. And therefore, and these were not available, these species were not available in our Indian gene bank I mentioned before. We have one. And so I imported, 12 species and total about 44 accessions of these 12 species for wheat. And this is the list, by the way, which is from the old records, sometimes in 1985. And using all this, for my PhD, then I could discover some new sources of resistance, particularly to drought tolerance in wheat, in terms of, for example, grain yield stability in Triticum spherococcum as one of the species, and Triticum vevilloi, the other one, vevilloi, the other one. Earlier, we were only depending on Triticum mistivum as particular cultivar C306. So in addition to this, we were able to identify 2 more species. And these were a couple of papers published in New Phytica where details are already given. But then, as I said before, that I wanted to achieve something big. And I wanted to also change myself with changing times. So what I did was, I was looking badly for an opportunity, and I worked hard for it to get an opportunity to work in one of the best universities of the world. And fortunately, I got an opportunity to work at Harvard University, where Professor Lawrence Bogored was trying to figure out, that there are certain photosynthetic genes, the proteins of which, photosynthetic proteins, they express in either mesophyll cells. I'm talking of C4 maize leaf section, it is shown here. they either express in mesophyll cells or they express only in bundle sheath cells. These are the bundle sheath cells. And this is the vascular bundle in between. Around these are bundle sheath cells. And around these bundle sheath cells, you have a layer of mesophyll cells. So the idea was to know that what regulates the expression of some of these photosynthetic genes in particular cell type, whether in mesophils or in bundle sheath cells. So we cloned certain genes. For example, here, chlorophyll AB binding protein gene, whole gene was cloned, but then we took the promoter thinking that it is a transcriptional level regulation which must be taking place and to express these genes only in particular cell type. So one gene of the protein of which was always in mesophyll cells, we took a promoter of this gene, hooked it in front of Gus, in front of it as a reporter gene. Similarly, the smaller subunit of Ribusco, which will always be present and found in bundle sheath cells, we took a promoter, of that, and to study the reporter using reporter gene as Gus, its expression, through bombardment, as a transient expression assay. So the assay we developed it first. It was not available that time. So we took these upstream regulatory sequences of CAB1, CABM1 from maize, and RBCS, again from maize. And we saw that if you use a promoter region or the upstream regulatory sequence of this particular gene, CABM1, you find the expression remains confined only to mesophyll cells of this. You don't see any expression in bundle sheath cells. So that means the regulation is at the transcription level, okay? Similarly, again, a beautiful result we got that the regulation is also at the transcription level when you use the promoter region of RBCS gene, and you can see here expression remains confined to bundle sheath cells. So I'm trying to show you from here, these were my first lessons of plant molecular biology, and I was able to then learn the whole technology of isolation of genes and developing gene constructs and answering certain questions with regard to functional genomics or with regard to even transgenic crop development. So during that time, it was 1990 to 92. During that time, the birth of modern genetic modification had already taken place. Okay, and when I was at Harvard from this period to this period, during this period, in 1992, we came to know that flavor sour tomato, the genetically modified tomato with delayed ripening, became the first GM crop approved by the US Department of Agriculture. So when I was returning to India, obviously, the idea was again to venture something big. And if we can use this knowledge and training in entering the area of developing GM crops. But as you would have seen, my interest remained only on abiotic stress tolerance with my PhD experience of drought resistance working on wheat. And that time now going deeper into these, understanding the molecular mechanisms of drought tolerance or in general abiotic stress tolerance in crop plants. Then I came across in 1990 again, around that period of time, the first review article in Plant Cell, titled Gene Expression in Response to Abscisic Acid and Osmotic Stress, wherein they said that the several genes have been reported, which get induced as a result of these abiotic stresses, but their function remains unknown. So that time, the first immediate question, scientists were trying to ask that if you overexpress some of these unknown function genes, can we try to find out their function, or can we also try to, also produce plants with these genes? If you overexpress them in transgenic plants, these plants could be then having increased abiotic stress tolerance. So two-pronged approach. One is that you will know their function, and if you're successful in developing transgenic plants in an agronomically important crop, then you will have the genetically modified crops with improved function or improved tolerance to abiotic stresses. So I picked up one such gene with unknown function, and that was this Osmotin gene. Osmotin gene, that time, only protein, lot of information was available that it is associated with osmotic stress adaptation in plant cells. It accumulates up to 12% of total cell protein, which is quite large in quantity, in cells adapted to grow in medium with low water potential, and its messenger RNA synthesis are further induced by all these kind of abiotic stresses like ABA, salt, and drought stress. So this was an attractive attractive proposal, it appeared to me, that can we try to work on osmotin, and our goal remained functional characterization of osmotin by overexpression in transgenic plants. So we developed this gene construct, just to show you the students, This is nothing new at the moment right now, but I'm just trying to traverse through that history through which I had gone through, and just to inspire the students. Then we developed this construct, with 35S promoter, which is a constitutive promoter, and a terminator. And then we started developing transgenic plants to see whether it's able to impart tolerance to abiotic stresses or not in crops like tobacco, potato, tomato, and mustard. That was the first journey I started with. And in fact, again, we took up a challenge of developing transgenic potato by one of my master students, Ms. Komal Mathur, who is currently a professor in one of the private universities. Master's thesis in 1995 to 97, talking of transgenic potato was considered big, I believe. And we were successful in developing this transgenic potato with Osmotin gene. And also we then developed a protocol for developing transgenic tomato. As you can see here, a foreign gene insertion through agrobacteria mediated transformation, and then you having green shoots coming up, under selection, kanamycin, and then further, these green shoots growing, and you then allow them to root, and then you transfer them, to a pot in soil, and then you see that these plants grow, they flower and produce fruits. And then, of course, you do this molecular analysis. So this is something we did after potato and tobacco, and developed transgenic tomatoes with Osmotin gene. And we could find out that Osmotin gene is able to provide tolerance to, to this tomato, both under salinity stress as well as the drought stress. And we could then verify these results as well using southern analysis, which was very hard that time to achieve good results, good blot like this. And then further we, studied the expression of these genes, and then this is using northern analysis, we were able to express, and see the expression of at least 2 of those genes out of many which we found positive for southern and northern. We see that they are able to express beautifully well. And then we continued with our research using Osmotin overexpression in mustard. And again, we found that it is able to impart tolerance to NaCl, as you can see here, to the mustard plants as well. And both, again, salinity stress as well as it is doing better under drought stress. When you can withhold watering, for about 5 days, you see that these transgenic plants, they're doing better than the wild type or the non-transgenics. So we continued further, as per the regulatory system of Government of India, that we undertook event selection trial at IARI fields, and this is, you can say, contained field trial. We did it, and we did it actually with both tomato and mustard. And what you see here is the RCGM contained field trial of Osmotin mustard, event selection. Here on the lower panel, you see this is an irrigated field, normal irrigation, so as to compare, as an irrigated control with non-irrigated, as you can see in the upper panel. You don't see any kind of irrigation except one irrigation, we gave. And then we found that some of the events doing much better, than the wild type. These 2 events, we were able to select. And then we got approval again from both RCGM and GEAC. RCGM is Review Committee on Genetic Manipulation, followed by Genetic Engineering Appraisal Committee, which are 2 important committees to give you approval for doing your work on recombinant technology related work, be it in crops or in pharma sector as well. So we got approval for the event selection first, and then followed by biosafety research level trial 1, followed by with Osmotin mustard, and then we found good results there as well. So then actually what we observed, because our objective was to study the function of this Osmotin gene, and the possible role of Osmotin imparting tolerance to abiotic stresses was through retaining chlorophyll or proline accumulation, or higher leaf photosynthesis, preventing the accumulation of reactive oxygen species, or increase in relative water content, which is very important and a very simple tool to measure, the status, water status of the leaves, particularly under stress conditions, and all these contributed to improved plant growth. Then, important was to again, share this now Osmotin gene construct, because during that time, I'm talking of, let's say 1995 to 2000 or so, we did not have many gene constructs, available in the country in various labs. And since I had developed this construct and I had tested it already in a few of the crops, I thought, as there was a demand as well from various other labs in the country, I started sharing it, and my daughter teaches me sharing is caring. So I started sharing it with all those who wanted it. And I'm happy today, that I shared with National Bureau of Plant Genetic Resources. And based on this, I shared my transgenic tomato with Osmotin gene. And based on this availability of that material, they were able to establish or strengthen further the GM detection lab. I shared with one of the big institutes, Indian Institute of Genetic Integrated Genomics Biology Institute, or CSIR, where they were able to strengthen their biosafety lab. Or in some other crops, they were used, for example, in tea, tomatoes, strawberry, mustard, rubber, capsicum, rice, by different institutions in the country or universities. Just to show you some of the examples, this is that Institute of Genomics and Integrative Biology. Dr. Naveen Arora, who was already working on these identification of allergens or purification of recombinant allergens and safety evaluation of genetically modified crops. So he was able to use, the gene construct which we prepared for many of his studies, and he could further strengthen his laboratory. Similarly, Indian Institute of Himalayan Resources Technology, in Palampur, was looking for a gene for developing transgenic tea plants, and they were able to develop transgenic tea. It did not only have increased stress tolerance, also it got higher quality product as a result of overexpression of Osmotin gene. And then DRDO is one of our, defense lab, the Defense Research and Development Organization, one of the institutions, they developed Osmotin transgenic tomato, and they found that this is able to behave, better, much better under cold stress. You can see these transgenics than the wild types, and all these led to, lot of good publications in different journals. All publications, whatever the work, we did with Osmotin gene, not only from my own lab, but I'm talking of from all of the labs where I shared this gene, we were able to have many good publications. But then from here, again, something big was there in our thinking process, that now what at the national level can we have a big project on developing transgenic crops, and which we succeeded that time in getting a project for roughly rupees 100 crores, from Indian currency point of view. And it was, I think, sometimes in, again, 1995, and, during that time, that we were able to develop a big project first time on developing transgenic in crops for different traits. We call it as ICAR Network Project on Transgenics in Crops, NPTC, and the idea was to develop some of these transgenics as listed here for different traits like insect pest, resistance to insect pest, or resistance to drought or salinity stress, resistance to viruses, or to different traits. And my responsibility there was, apart from coordinating the transgenic component of this particular project, to also clone genes and promoters for abiotic stress tolerance, because we had a mechanism that some of the institutions who were already doing that kind of work of cloning the genes, developing gene constructs, that they will be able to develop those genes and gene constructs more, and will be able to provide across to our partners, those who have the expertise of developing transgenic crops. So we developed gene constructs for many of these crops by cloning genes and promoters from various sources. And the genes, again, you will see we clone from various plant genetic resources, different species, or wild species, which would have got that better capacity to provide tolerance. So we cloned several of these late embryogenesis abundant protein genes, LIA genes, from some of these brassica species, or also from one of the genotypes highly drought tolerant I mentioned before in wheat, that is C306, and different kinds of category of these genes. So one of the genes from Brassica napus, LIA category 4, had not been studied for its functional analysis. So we found that when you overexpress this in Rhabdopsis, we find that these transgenics, they recover better. These are the 3 transgenics here, with constitutive promoter, and these are with abiotic stress-inducible promoter, the 3 transgenics. So compared to wild type, these transgenics, they recover better, as you can see after 1 week of, recovery, and compared to the wild type with either of the promoter. And later, this role of group LEA4 genes in abiotic stress tolerance was established by other groups in different other crops. And we also cloned the LEA4 promoter, and which was again used as a stress-inducible promoter, to drive the expression of various other, structural genes in developing transgenics for improved tolerance to abiotic stresses. So this is one of the promoters, other than the LEA promoter. I'm talking about MIP02 promoter from rice, landrace, Nagina 22. We did a functional analysis of this to see that it is really induced by, by particularly, let's say, drought and salt, but this particular deletion we were trying to do was not induced in cold, so we could identify, which is that portion of the promoter which is responsible for driving expression in, when it is induced either by drought, salt, or cold. Of course, this work has not been published yet. So now with the availability of the genes and different promoters, we developed 2 different gene constructs. One with RD29A promoter driving expression of DREB. This is the drought responsible element binding protein, 1A. It's been used by several other labs. And the other one we use, this is zinc finger. Actually, this was earlier reported in our dropsies as JAD12, but this is zinc finger protein. We call it as BCZF1 actually, and we used the promoter here, as I said before, LEA4 promoter, from Brassica carinata or Napus. So these 2 constructs we developed, and our idea was to spread over, through that network mode to all other institutions who were otherwise ready for their transformation protocol and looking for some of these genes and gene constructs to develop transgenic crops. So we shared these 2 gene constructs, with, we call it as National Agricultural Research System, comprising, these are some of the ICAR institutes or some of the state agriculture universities. These are 2 different state agriculture universities. And these are the ICAR institutes working, for example, this is cotton, this is vegetables, this is groundnut, this is tomato, this is pulses, this is rice, and this is also rice. So we developed several of these crops, and my responsibility was also to continue developing transgenic mustard. So we developed transgenic mustard with Osmotin, I mentioned before, with this Brassica carinata ZF1. Okay, we also had availability of OTS-BA for trehalose biosynthesis and also DREB1A. So Osmotin, we had with these 4 different genes, we through cross hybridization, again, we started pyramiding some of the genes, and we were able to have this, 2 to 3 genes pyramided together in one particular genetic background for enhanced tolerance to abiotic stresses. Okay, and then the other labs, I said in the National Agricultural System, for example, they developed transgenic wheat, rice, 3 different or 4 different labs, mustard, chickpea, groundnut, tomato, again tomato with another gene, and then cotton and sugarcane. So these were all developed by various institutions, as you can see here, and this is the reference, it's all published. And more number of publications came out of the use of these 2 different genes and gene constructs by various partners in our network program. So finally, we have some of these crops which are in pipeline today for regulatory approval. For example, we have transgenic groundnut with DREB1A, transgenic mustard with BCZ12 or BCZF1, actually, I keep repeating that. And then we have transgenic chickpea with DREB1A, again, rice, DREB1A, 2 different kinds, 2 different labs, and 2 different varieties, actually. And then we have sugarcane with BCZ12 or ZF1, tomato with both, Z12, ZF1, and DREB1A. And this is the, reproductive stage of these same transgenic potatoes. And all this has been published. And now these are some of the selected events I'm just trying to show you here, which will go through the regulatory approval, I believe, soon. But then, when we talk of genome engineering, today we're talking of genome editing, all right, yes, important. But then we also should know more about chloroplast genome engineering. Before we got success in targeting this transgene integration, otherwise, or de novo, some kind of, substitutions, in vivo system, we have this chloroplast genetic engineering, which is very, very important and a very versatile tool. And my interest was, as a Rockefeller Biotechnology Fellow, I had an opportunity to work with Dr. Pal Maliga, one of the world authorities on chloroplast transformation, and working at Rutgers University, and we had an opportunity to work with him and learn about this technology. So when I came back India again, we wanted to, actually speaking, we were more excited. Sometimes again, in the quest of making big achievements, you fail. And actually I started, after I came back, from Rutgers University, we started in maize. And one of my bright students from Iran, Mr. Baghban Ruz, who is now associate professor in Tabriz University in Iran, actually 20 years ago when he was in my lab, we struggled hard for 4 to 5 years continuously to develop this chloroplast transformation technology in maize. And as Dr. Ruz, he was struggling that time. He had a lot of good experience of, transformation of maize, but we could not succeed simply because the spectramycin resistant gene, which we were trying to use, turned out to be having its own inbuilt kind of resistance. We could not do kind of a good selection using, the selectable marker gene that time, not too much of diversity available for these selectable marker genes. We had only AADA imparting resistance to spectramycin and streptomycin, and it turned out that the maize had some kind of intrinsic, resistance to some of these antibiotic agents. And also, of course, this was a very great difficulty that there was no protocol of transformation from maize leaf tissues. We wanted it from maize tissues when you talk of chloroplast, though they're there on all the cells in different forms. But anyway, finally we succeeded in doing this, developing this protocol in eggplant, which is, brinjal, in Solanum melonginella. And in here, in chloroplast transformation, again, the idea is, if you know the whole sequence, of course, which we know now in so many number of species, the whole genome sequence of a particular crop, then you can target it, in the intragenic region based on homologous recombination, this kind of foreign gene integration. And now, actually speaking, Dr. Ralph Bock in Max Planck Institute in Germany, who was also working partly with Dr. Pal Maliga, is another world authority on chloroplast transformation. And he is given, very good leads available for taking this technology forward, not in the agriculture sector, also in the pharma sector. And, so what is important for India is that we should develop this technology, for those who are really interested in making, kind of a big difference. We must try to develop this technology in chickpea and pigeon pea. Of course, in rice and wheat, there are some reports. Here, there are no reports at all in these two crops. Rice, there are some reports already, but again, we need to be more successful there. In wheat is important, required. Maize, mustard, tomato, and potato, of course, these are already there, but we need to use them more for various traits. And what is the importance I'm talking about? I highlighted chickpea and pigeon pea, simply because you need durable resistance against insect pests, particularly, using chickpea, BT gene, native BT gene. Native BT gene, we need, through this technology, there's a possibility that you can have a full crop protection from an insect pest by expressing long double-stranded RNA in the plastids. This example shown here in potato this is Ralph Bock I was talking about. This is the wild type, and this is when you express actin gene, antisense actin gene in a nuclear background, and this is when you put this long double-stranded RNA, antisense, this is a long double-stranded RNA when you put, when you express in nucleus versus when you express in chloroplast. You can see here in chloroplast when it is expressed, long double-stranded RNA to the tune of up to 200 MER bases, you find there's a complete, protection almost here. And this is using the actin gene of Colorado potato beetle, beta-actin gene of this Colorado potato beetle, which is not a problem, of course, this beetle, but it can be utilized for other pests, which are there in India, and it could be particularly utilized for chickpea and pigeon pea, even for brinjal, for that matter. We have developed this protocol, to have, durable resistance against some of these insect pest pressures. So this is the beauty of this technology you have. And I always keep promoting, actually speaking, GM crops to drive benefit to the farmers. And this is out of my recent article. And I like playing cricket. I have played a lot of cricket. Actually could not make big in cricket, but then, I have my own cricket 11, I call it, +1 as an extra of some of these GM crops for India. We need, after BT cotton is the only crop commercialized, we need BT brinjal. We need GM mustard, which Professor Deepak Pental has developed, hybrid mustard, which showing about 25 to 30% enhancement in yield. We need HT BT cotton, herbicide tolerance BT cotton, nitrogen use efficient cotton, drought and salt tolerant cotton. I'm talking of sugarcane, which has been developed with the gene I gave an example before. HT BT maize. Then we need BT chickpea and BT pigeon pea, preferably if somebody can do it through chloroplast transformation. DREB1A rice, which has already been developed by various labs. DREB1 chickpea, which has been developed by Indian Institute of Pulses, research in Kanpur. And late blight resistant potato, excellent work done by Central Potato Research Institute, Shimla. So this is something India needs immediately. While we have 20 to 30 more, with regard to nutrition also we need, which is not listed here, some zinc and iron, enriched rice developed by Bose Institute with Professor Sopan Datta, and many other labs. So we have more of such transgenics already available, but we need to, actually speaking, bring them back to the farmer's field. So I said, this is my journey. I have talked to you about little bit only about germplasm. To start with, during my PhD program, I imported some germplasm I mentioned. Then I talked to you about genes and GM crops, but then we always come back to germplasm for two more reasons now. By this time, I had known enough that germplasm, we need to understand as a national property we have of our own, material which is available in India, which belongs to India, because we have now Convention on Biological Diversity in 1993, it came into being, followed by International Treaty on Plant Genetic Resources, Food and Agriculture to promote sharing of this germplasm through various regulatory means and principles. So, keeping that in view, what germplasm we have in India, how much value we can find in it, and how can we use more and more for genome editing now. And its importance has increased further now because of genome editing, simply because what is genome editing? Genome editing actually is an ability to edit native crop genes coding for important traits. So until unless you know what are those native crop genes to be mutated or to be edited, we will not be able to make much headway with genome editing. While of course there's already a lot of information available, many transgenics in different crops, more than 20 crops, people are working mostly in rice. But coming to the Indian scenario, we need to understand our germplasm better. With regard to keeping in view the Convention on Biological Diversity, what it is all about. It is dedicated, the message here from CBD is dedicated to promoting sustainable development. It is about people and our need for food security, medicines, fresh air and water, shelter, and a clean and healthy environment in which we live. So, and biological diversity, I'm not talking only about crops. I said even before, the diversity, to start with, is important in different organisms. So therefore, what are the objectives now, laid out for CBD is the conservation of this biological diversity. I mentioned about little loss of diversity in the beginning when I started. The idea here is now we must conserve as much as possible, okay? And we must use it sustainably. It's all different components, and fair and equitable sharing of benefits arising out of utilization of genetic resources with farmers who have been conserving it for centuries, from where only the scientists collected, of course, they were collected even from the wild habitats, but then some of the farmers, custodian farmers, they've been conserving it for centuries, so there could be a fair and equitable sharing if the scientists use those genetic resources and tomorrow they commercialize it, and some kind of benefits must go back to that country as a matter of fact, because CBD allows each country to have its sovereign right over all such biological diversity they possess in their own, kind of geographical jurisdiction. So India is a signatory to this Convention on Biological Diversity. So therefore, plant germplasm, when I'm talking about, assumes even further significance. And globally, there's a lot of significance attached, to these conservation of plant genetic resources in international gene banks. There are about 1,750 gene banks in the world, conserving 7.4 million, 74 lakhs accessions, means different lines of different crops, and out of these number of gene banks, the majority of gene banks actually are where there's large amount of germplasm present are with CG center, Consultative Group International Research Center. For example, we have here in India, ICRISET, we have ICARDA in Syria. Now it's of course moved to Jordan and Morocco. Then we have, this is basically now Biodiversity International in Rome. It's an alliance of Biodiversity International and CIET. Then we have CIMET in Mexico. And then we have, IRI rice in Manila. On potato, CIP, Central Institute, International Potato Center in Lima. And we got CIET, I said before, which is now combination in alliance with Biodiversity International. So some of these are international gene banks holding a large, germplasm accessions of various crops, or crop-specific institutions. But just for the information of the audience, apart from some of these gene banks as shown here on the global map, there is one, you can see here is a dot near North Pole. We call it as Svalbard Global Seed Vault. This is unique in itself because this is an international gene bank, to conserve the plant genetic resources forever. Okay, anyone who wants to deposit is free to deposit the material here as a black box arrangement that nobody will use it until they can only draw it whenever they feel like, using it, getting it from there. So while again thinking a little big here, I was interested that we must try to make our presence felt there as well in terms of depositing some of the Indian germplasm accessions, all through the official procedure in this global permafrost facility, which is conserving PGR, I said before, forever. And this is a unique facility, as you can see here, you just don't see other than sea and snow all around. And this is that, Svalbard Global Seed Vault. This is a tunnel actually, it goes under the, rock in the hills. And this is inside, it's all, frost temperature, quite low. And I was happy to, be a part of this delegation. When I took this material from India, you can see Indian flag, it is stored here in one of the racks, where there's many other countries have got it. And we visited this, in April 2014. But then in coming back home, we have our own national gene bank, and which is second largest gene bank in the world. So, it was third gene bank earlier after US and China, but another big achievement, I would say from Indian point of view, that we were able to make it second largest today now with about 4.5, lakhs accessions, 0.45 million accessions conserved, in this national gene bank, of all different, crop species we're talking about for food and agriculture, particularly talking food and agriculture. And this is, the efforts being appreciated by Dr. Swaminathan, Professor Sopandatta, many others you can see, including Mary Haga, who was one time, minister in Norway, government Norway, but then she was also executive director when she was state of GCDT, which is a global crop diversity trust in Bonn, Germany. And our honorable ministers also, they have, a lot of interest in maintaining, these kind of facilities we have of the international stature. But then not only that you keep these germplasm conserved, which is very important task by itself in these national gene banks or international gene banks, but then, as I said before, if you have to do genome editing, or even if you have to do genetic engineering, you need novel genes and gene combinations and alleles. So for searching these genes, you depend back again all the time on these plant genetic resources. Okay, it was way back in 1937, one of our stalwarts, Dr. B.P. Paul, he, in his paper said the search for new genes, he advocated a systematic conservation, evaluation, and use of genetic variability, which of course was then used by Dr. Norman Borlaug for wheat, which gave rise to green revolution. But again, he kept talking, that what is important is to identify some of these new material and incorporating it into our breeding programs. But then I said before, if you go back and see, this material runs into thousands in number, thousands. For example, here in cereals, we have got rice, which is 105,000. Wheat, we have about 25 to 30,000. Grain legumes, I'm talking about all oil seeds, chickpea, pigeon pea, and others, we've got 67,000. Oil seeds, 60,000. So what I'm saying is it's running into thousands. So how can you use thousands together in a breeding program? It's not possible. So one of the greatest scientists, again, historically speaking, Otto Frankel, he developed a concept called as developing a core set that if you got 30,000, you develop 10% of this entire collection, as a core set, which will be only 3,000. And the 3,000 is something, a small number, manageable enough for use by various approaches, So we took up that kind of a challenge, with wheat, and this was something, the biggest experiment I could ever imagine in my lifetime. And I'm happy I was associated with it. And this was characterization of the entire wheat germplasm from the Indian National Gene Bank to develop a core, and we have in India about 25,000 such gene accessions, number of accessions, and which was a hard task to take them all out, from a gene bank and characterize them by growing them in one of the best fields all together at one site, which was really speaking a gigantic task. But, fortunately we were able to do it, and we could find a lot of variability, in terms, you can see your plant types, or even chlorophyll content or growth habits. It is kind of spreading in nature. This is more erect in nature. This is higher in chlorophyll. You can see totally dark. This is very less in chlorophyll. And this is another accession, which has got, very high amount of leaf epicuticular wax, compared to one which is very less. And you can see here the leaves are quite broad, as compared to many of these which are narrow. So there's a lot of genetic variability. And this is what is the repertoire of those genes we require today for climate change and many other traits. So finally, I'm cutting the story short, of course. We developed a core set, and core set contained about 2,000 accessions out of 22,000 accessions. Now, what is the advantage of this core set here is, I said 10% of this total number, and it actually speaking, contains about 90% of total genetic variability. Whatever the genetic variability is, let's say 100%, we have an entire collection from the Indian Gene Bank point of view. Out of this, more than around 90% of variability we still have in these core sets, which is only about 2,000 accessions. And we finally, of course, developed a core set of a composite core set, different species, Triticum mistivum, Triticum dicoccum, Triticum durum, all together. And this is recently published in Crop Science. And then we found new sources of resistance to disease against rust and spot blotch, from these many number of accessions. We found new sources of resistance to powdery mildew, again from the same number of accessions grown in different hotspots. I'm not going into the details of those. And then I was so happy, that our efforts were recognized by Crop Science Journal, which was released on November 11, 2020, and it appeared, our effort appeared on the cover of the Crop Science Journal, where they referred, gave a reference to both of our papers, that we published on wheat. And we have now, keeping climate change in view, we have developed a reference set for terminal heat tolerance in wheat with about 3,000 accessions based on certain parameters, which are all associated closely, with heat tolerance. So we have this material as well. And our study, you can see here, this is a global wheat director from CIMMYT, and some of the scientists from India. You can see here, they all, gathered here, including Dr. Vishwanathan, who is a leading scientist we have in IARI, working as the head of the division plant physiology. And important was, actually speaking, when we thought we should try to do this experiment of developing core and take out all that material. But when you do an experiment, you need little, quantity of material, which is minimal. You need it always. We conserve very little material in our gene bank. So when we took this initiative, we found that we have not enough material of these 16,000 accessions out of about 25 to 30. Of course, there were duplicates, which we removed as well. So we actually multiplied as an off-season, experiment in Wellington, in Tamil Nadu, and which again became, kind of good information for media, and they covered it. That such a experiment was conducted for the first time, by any Indian institution. And of course, then it attracted Limca Book of Records. It was recorded as one of the largest experiments. And Times of India recently, again, November 26, 2000, came up with this news item, that understanding wheat like never before. And one of the, again, great scientists, plant physiologist, working on physiological breeding at CIMMYT, and very great interest in terminal heat stress tolerance, Dr. Matthew Reynolds, he specifically came down, traveled, You know, it's a long journey, you know, from Mexico to India. He traveled just for 2 to 3 days and to look at our experiment and have discussion with us. And I was so pleased. And these are, you know, our Indian expert. This is Professor Deepak Pental and many others who visited, you know, the farm to see this experiment. And I'm very happy to show you this slide, actually speaking, you know, thanks to WhatsApp, that I shared this information with them. And I got these messages from some of these top leaders of agriculture or agricultural sciences in India. To start with, Dr. Talochan Mahapatra, who is today the Secretary of Department of Agriculture, Research and Education, Government of India, and Director General of Indian Council of Agricultural Research. He said, excellent, it's a milestone, a landmark contribution. You know, you feel happy about it. Professor Deepak Pental, the former Vice Chancellor of Delhi University, says it's a great piece of work. Nobody in India has dared to undertake such an activity on germplasm. You know, then Professor Sudhir Sapori, you know, he is again one of the greatest scientists, plant scientists, you know, and who was earlier the Vice Chancellor of JNU, now working in ICGEB. He said this is the result of your commitment and passion for doing good science with application in our agriculture. Then we have actually, you know, one technical expert, but he is an IAS and who happens to be country director of ICRISAT, Dr. Padhi. He says, very proud to know about your work that would positively contribute in development of climate resilient and disease resistant and tolerant wheat varieties. And currently the director, a very noted plant breeder of this country, you know, Dr. Ashok Singh says, indeed an historical paper, I will circulate among the colleagues so as to use this material. He is the rice breeder himself. And I was very happy. And Professor Dr. Talochan Mahapatra, as you all know, is a world-renowned scientist in the area of genomics. So this is something you feel happy and proud, you know, when you get such nice comments. And more importantly, again, you know, Dr. Swaminathan, when he came to know, he visited, of course, National Bureau of Plant Genetic Resources, sent me an email message, you know, and he says, I was happy to visit NBPGR. I was particularly happy that converting NBPGR as a functional conservation center with the kind of experiment I'm talking about, which we did with wheat. Not only with wheat, we did it also with chickpea, but I'm not having enough time to go through with the chickpea experiment. But then by this time, 2013, 14, 15, genome editing came into picture. And again, germplasm, I said before, will ever remain important, not only for all this national, international point of view, conserving sustainable use, utilization for crop improvement by plant breeders, but also by the genome editing, you know, experts. You know, as I said before, genome editing will require information on what are those plant genes which we need to mutate, you know, and bring back the genes which are lost from wild species to the cultivated, you know, genetic background. So I'm just trying to show you here, you know, one of our genes which we cloned, which later turned out to be an ABA receptor. Actually, ABA, abscisic acid, is a versatile hormone. You know, it has a diverse role to play in plant stress particularly, and also for plant development under stress conditions. So, you know, until 2009, ABA receptors were not discovered. So one of the science breakthroughs of 2009 in top 10 was discovery of these ABA receptors. Okay, and once you have these ABA receptors known, you know, it's, I don't have time to go through all of it with you. You know, there are about 12 as shown here. You know, 12 ABA receptors in rice, or is a stiva, you know, in rice. So actually, one of the discoverers was our collaborators. You know, Dr. Vishwanathan, again, I mentioned before, who worked with Professor Jiakang Zhu, and they, you know, discovered, the two groups discovered these ABA receptors, one from Germany, another from Dr. Jiakang Zhu's lab, you know, and they did it. And then they cloned then all those ABA, you know, abscisic acid reporter genes, receptor genes from, you know, drought tolerant. Dr. Vishwanathan had done it from rice cultivar or landrace, again at 22, you know. They have all been done now, actually. But in 2009, October, you know, before even ABA receptors were discovered, you know, luckily we had cloned one gene, PIL3. You know, we found it, of course, it was published in 2020, but in 2009, we found that, yes, we cloned this PIL3 gene, now renamed as PIL10, which was the first functionally characterized ABA receptor from a monocot rice landrace, again at 22. And we could see that it is able to impart tolerance, you know, to cold stress. This is the wild type, wild type, of course, without stress, you can see here. And when you overexpress this particular gene, candidate gene in rhodopsis, you find roots are better and shoots are growing better under cold stress. And then later, it was verified by Dr. Viswanathan's group at IARI that overexpression of this ABA receptor PIL10 gene confers drought and cold tolerance to indica rice as well, which we had, found it in rhodopsis. And then using genome editing tools, this is one of the landmark work I have come across on genome editing for increased rice growth and yield by editing the ABA receptors. I said before there are about 12 ABA receptors, and Dr. Jiakang Zhu, who was one of those two groups to discover ABA receptors, they developed this transgenic rice, with permutation combinations of all ABA receptors. And they found the one with like 1, 2, 3, 4, 5, and 6, when they mutate, the plants grow better, as compared to wild type under field conditions, in fact. And out of these 6, when they picked up 3, 1, 4, and 6, they even turned out to be better than the combination of these 6 receptor genes, And here you can see the yield per plot. In wild type, if it was about 1,500 gram per plot, it got up to 2,000 roughly, gram per plot with these mutations of 3 ABA receptors, 1, 4, and 6, 30 to 35% enhanced yield. So actually speaking, earlier it was known that it only can protect or make the plant survive under given abiotic stress or drought stress conditions. But here it finds that even if you mutate, some of the genes out of 12, they have got even better growth and better yield, under field conditions. So there is a balance between growth and stress adaptation, which was new and discovered by Professor Jiakang Zhu's group. But then genome editing, of course, continues to be important. And I highlighted this, that we should try to make use of this technology, particularly for developing climate resilient crops, and there are different traits which we would like to use for some of these climate, resilient crops. For example, you can see here, tolerance to drought, enhanced flooding tolerance, improved nutrient exhaustion, radiation efficiency, improved pest and pathogen resistance, symbiotic nitrogen fixation, etc. There are several traits which we can try to utilize. And with regard to regulatory landscape, many of these countries shown in green, they have already said it should be treated as non-GMO. The plants, particularly, some of those there are 3 different categories: SDN1, SDN2, and SDN3. SDN1 and 2, where the plant's own genes are mutated, those are treated as non-GMO from the regulatory perspective of some of these countries like Canada, US, Australia, Japan, Israel, and India, and many other countries. The discussions are still going on. But we had a discussion in a National Academy of Agricultural Sciences, and where we feel that, GM crops or genome-edited crops to be integral component of this self-reliant India. And for this, we need a clear policy. And we propose that SDN1 and SDN2 type of products without foreign DNA to be treated as non-GMO, because these genome-edited regulatory guidelines are still not official from Government of India. So we had our own national consultation by this academy, and we propose, to be treated, at least these 2 categories without foreign DNA, to be treated as non-GMO. And what I feel that we need political support more and more in favor and relevant, in favor of relevant GM, genetically modified or genome edited crop plants, which are important for sustainability and profitability of smallholder farmers. So with this, friends, thank you. And I acknowledge the help of my institutions where I was working, my collaborators, Dr. K.V. Prabhu, who is currently the chairperson of PPFRA, Dr. A.K. Singh, Director IRI, Dr. D.K. Yadav, Dr. Viswanathan, and those NPTC partners, scientists of NBPGR, my students, many of them. And of course, another big thing for the students' point of view, I'm happy that we were able to launch in India one of its own kind of project. We call it as National Agricultural Higher Education Project, National Agricultural Higher Education Project, NAHEP, which was launched in July 2017, and which is about rupees 1,100 crores. And with this, a big thanks to you all. And this is only a, kind of a compilation I'm showing you of wild relatives, of cultivated plants, which came in 2014, which is all this information, various attributes, whatever the cultivated relatives we have of our cultivated plants in India. So with this, thanks to the Bioingene team, and a big thanks to all the viewers, all the participants, for their time. And if there are certain questions, of course, I'll be very happy to take up those. And thank you, Jacinta, for the introduction, and Mrs. and Mr. Majumdar for organizing this. Thank you very much.
Thank you, sir.
Thank you, sir.
Thank you. Sir, now you can stop your screen share, and I will request Jacinta to go for the question answer round. And we have already few questions, and we go first with that, and we're still collecting the questions from you to live chat. Those are not answered, we will answer after the webinar with the consent of our Bansal sir. We will send the all answer to the attendees and those have asked the question before and this live chat. So Jacinta, go for the question answer round. You ask the question directly from the list.
Thanks, Shivo. And thanks again for the great presentation.
Thank you.
So these are questions that we've been receiving in the last couple of weeks through the website. The first question, so I've tried to order them and group them together. The first section relates to the safety of GM crops. And the first question was by far the most asked question. Are GM crops safe for human consumption?
The short answer is yes, very much they are safe.
Okay, great.
And why do I say so?
Yep.
Because there have been tests done over last 20 years, more than 20 years, and these crops have been consumed by, let's say, the biggest consumers, number of consumers are in the United States of America, and there is no such reported case of any untoward incident with regard to consumption of GM crops. So they are all very safe. Not only they are being tested over and again whenever before they are commercialized, and they're all very safe, actually speaking.
Yeah. So some follow-up questions from other Bioingene participants are, how are these potential risks determined? And there was a question from Merlin in the Philippines, who was quite specific in his question, asking on how do we do risk assessments of GM crops if environmental activists are opposing the establishment of these tests?
You're right. In fact, I will come to that part from the Philippines later. But before that, I would like to say that the risk assessment and all such analysis are done by all these various experiments as advised by these guidelines under the regulation by different national governments. We have, for example, toxicity analysis is done, allergenicity tests are done, soil microbes are being studied wherever the GM crops are grown. Before they are even released, I'm talking about all these tests are done. Under contained field trial, there are a whole list of tests which are to be performed at various levels. For example, in India, we carry out these biosafety research trial 1, biosafety research level trial 2, and wherein all those data, are recorded and they are, then submitted to this review committee on genetic manipulation, and which is composed of about 20 to 30 experts in various fields, including even medical doctors. So there is a thorough discussion on those data, and if any such information is obtained, which is not carried forward, that material is never carried forward, But and therefore there are various means very well worked out at national international level as well, to carry out all these tests and find that they are safe, only then they are taken forward. Now, with regard to the question from Philippines, there are of course environmentalists who are against it, because everybody has, a freedom to convey their feelings and messages, very right. And only thing is, that these tests otherwise are being done, as I said before, in some of these laboratory tests or in the greenhouse material, or even under contained and confined field trials. So I would only request the environmentalists to have a discussion, actually speaking, that we should try to discuss out what their concerns are, and we should as scientists try to address all those concerns and find if there is any meaningful information coming up, we should try to always incorporate into our further experiments, to see that the technology is taken ahead and used in a very responsible manner, which has been done so far, I'm sure, so good, particularly with GM crops.
Great, thank you. Dr. Umabati Sahu from India asks, are there any environmental concerns regarding GM crops?
Well, environmental concerns could be, as we have been talking about it, that if there is any adverse impact on soil microbes, there's no such impact which has been noticed. Whether there is any kind of a loss of biodiversity, people are talking about it. There's no such loss of biodiversity taking place as a result of GM crops. I would even say in India, we have witnessed an increase in biodiversity, okay? And I give an example of BT cotton is the only crop we're growing in India. After the BT crop has been now growing for the last about, let's say 20 years, and in 2002, it came into certain region of India, it was introduced and released. But at least for the last about 15 years, we've been growing BT cotton. The number of honeybees, the population of honeybees in different parts of the country where BT cotton is grown has increased tremendously, actually. And the experiments have been done by some of the state agriculture universities. And another, environmental impact, people talk about it, whether, a gene from genetically modified crop could flow over to some of those non-related or wild species. In fact, that has also not been noticed at all. Even in BT brinjal, there were some, some concerns raised by some of the environmental scientists. But the point here, then it turned out from the experiments, otherwise, that, it's very hard, as I said before, to, crossbreed a wild species with a cultivated species. It's not easy. Even by human intervention, it doesn't happen. And even by human intervention, we are able to get F1 seed. It may not be turned out to be fertile. So it's not easy that, and also we must know any crop which is being commercialized and grown on large scale, which is genetically modified, and whether it has all its related species around it or not, really speaking. But no such concern has been, has at least come to my knowledge. I would even say with regard to biodiversity, it has even increased further with the use of GM crops. And there are many other advantages. Environmentally, it gives you clean environment. You spray less pesticide. How much pesticide has been reduced, the consumption of it wherever, these BT insect pest resistant crops are grown globally. And I saw one of the questions in the chat box, which, Mr. Majumdar had sent to me, was what are the demerits of GM crop? There was one question. And I thought that the demerit which came to my knowledge so far is that it is reducing the consumption of pesticides. Now if you take it as a demerit for those who are producing enough of pesticide for marketing, I would say it as a demerit of GM crop. That's the only demerit which I could think of at the moment.
Okay. Okay, so the next set of questions relates to the labeling of GM crops and products. Dr. Umabati Sahu from India asked, how can we as consumers differentiate GM vegetables from conventional vegetables?
Actually speaking, there are molecular tests, which we do all the time in the lab. And we know there is a specific gene, where it is located. We call it as an event. We characterize it, which gene, where it has got integrated onto the genome, how many copies of this are there, what is the level of expression, and what is that kind of an efficacy it provides to, or what is that kind of a better agronomic capacity or power it has given to the genetically modified plant. So these all molecular characterization are done in the lab, and before it is commercialized, all those agencies, regulatory agencies, that look into all that data. But once it has been deregulated, okay, and it is found acceptable for consumers, and it is found that it is not having any adverse impact on the environment or on biodiversity, etc., I believe, this is my, say here, according to me, that you don't need to have to worry. Though it is okay if you can afford to do, labeling. There's a few countries they're doing it, but then according to me, I think it's not required, really speaking, except that, if enough tests have been done and then if it is found safe, I think it's okay for me. Though we have in India a regulatory authority, we call it as FSSAI, Food Standards and Safety Authority of India, and they are looking into all those aspects as well. There is a scientific committee, and they're looking for it, in fact. But according to me as a scientist, for example, BT cotton is not labeled, okay? And people can argue that it is not consumed, but we have cotton seed oil, actually speaking, which is consumed. And if we have been consuming that for the last 15 years, I don't think it has caused any harm to anybody so far.
Great, thank you. So the next set of questions relates to the growing and handling of GM crops. Winma from the Philippines asks, do environmental factors affect the efficiency of GMOs? Why or how?
What kind of environmental factors? I don't know, somebody is talking about here, except that environmental factors, as we have seen in India, if we have got hybrids, I'm taking an example again of BT cotton, which are hybrids, which are being cultivated by the farmers, about 95% of adoption rate in India. And those BT cotton hybrids require more of naturally, resources to produce more because they give you higher yield. Not only because of BT cotton, even because there's a hybrid vigor, there's hybrid power. Without even BT, they would require more of resources. So, but in cotton, since we have this hybrid with BT gene, they would require more resources. And, in fact, there are areas where we don't have, for example, irrigation facility, so they would require more of irrigation, and to come up to that level of production, versus where it is all given full irrigation. So if you grow those GM crops under those environmental conditions, where not enough resources are available for the crop for it to be producing more, naturally it will show its impact otherwise, comparatively speaking. Yeah.
Cool. So Dr. Sithan from India asks, are there any differences in the nutrient requirements between traditional crops and GM crops?
No specific nutrient requirements, except I said again, just minutes ago I mentioned about more requirement of resources, but there's not specific that they will require specific nutrient. No, not really.
Okay. So we've got some questions related to GM technology. Brijesh Kumar from India asks, the advent of speed breeding has overcome the hurdle of long development cycles associated with traditional breeding. Do you think that GM crops will be accepted if there is an option of equally effective varieties developed through speed breeding?
In fact, we should, according to me, worry more about the product, okay? The product, which has all those attributes and characteristics which we require, for example, climate resilience, enhanced nutrition, and it comes through which route, we should not worry about it as much, okay? The route could be conventional breeding, route could be pre-breeding, route could be speed breeding, could be genetically modification or even genome editing, as long as the product is safe and has all those attributes we're looking forward to it, from the farmer's point of view, for sustainable agriculture, for the clean environment, we should accept it. So in a way, over the last 100 years or so, we have added such important technologies and tools, to the toolbox of plant breeders, that apart from conventional breeding, well, you have all these tools available as well. So which tool do you use? Which combination of tools you may like to use to develop a particular product, which allows you to develop that product? We should use all those products, actually speaking. Here, I would say we have a choice, So the onus is on the scientist now, which technology will allow us to develop, that kind of product which is badly needed. For example, BT cotton. You think BT cotton would have ever succeeded through speed breeding or through conventional plant breeding? Probably not. Therefore, it became a great success, and the gene from bacteria, which was otherwise used as a biopesticide to control a particular insect pest, now is there as an inbuilt resistance within the crop. So it became a successful case. And there are many other examples like this.
Great. Dr. Umesh Singh from India asks, what is the process of registering GM crops?
The process of registration of GM crops, that he should visit the website of Plant Protection Variety Farmers' Right Act, PPVFRA, where they are allowed to be registered as essentially derived varieties, EDVs we call them, essentially derived varieties. That varieties which are already there available, through breeding efforts, even commercialized. If in that variety you have added only a single gene for a particular trait, again, example of BT cotton, so that could be registered as the GM BT cotton variety with PPFRA as essentially derived variety. And there is Dr. KV Prabhu, I said before, as the chairperson, who himself is a very noted plant breeder, will be able to help him out a lot in this regard.
Great. So I've tried to combine a question from two participants. So Bim Meena and Mukesh Kumar from India ask a question along the lines of, why are transgenic crops, including cereals, oilseeds, and brinjal or eggplant, not allowed in India, while other developed countries are growing these transgenic crops?
Well, that's a question even I keep asking myself, and as I said before, each country is governed by an elected government, and they have their own social responsibility to carry forward, for the masses, so it is finally their decision. But as scientists, I would like to, in fact, urge and request, for example, Government of India in this case, that we must try to make use of all these advanced technologies as long as they are able to give us safe products. And so far, genetically modified crops through this rDNA technology has given very many safe products to many other countries. There's total about 70 countries who are consuming, not producing, who are consuming, genetically modified crop in one form or the other. However, there are about 29 countries which are growing these genetically modified crops, covering an area today, if I remember, about 190 million hectares, of at least a dozen of crops, and they find, that it is all safe for consumers. Even, for example, GM papaya, virus-resistant papaya, which is eaten raw, there's a less browning, apples, which are eaten raw, so I think we should try to see the safety point of view. They're all safe. Technology allows them, to be utilized and developed and utilized, for different traits, and it should be allowed. I would urge to the Government of India, and they are right, both of them, that yes, we should try to promote the use of technology. And not only this, even genome-edited crops should be, to some extent, even, remain free from regulation.
Yep, I agree. Okay, so this is the final question from the questions that we received in the last couple of weeks. And this is from another question that I've grouped from two different people. So Saeed Haidar from Pakistan and Merlin Buscato from the Philippines. Why are most people not interested in adopting GMO crops, and how can we help change people's perceptions?
Yeah, well, that's a good question. And I think that's what we scientists have been lacking in communicating, about the science in general, about the GM technology in particular, and what kind of advances are we making, and how safe are they in developing some of these genetically modified crops, and which are essential in nature to be utilized. I said in one of my articles recently that we need to increase the pace of generic development of these crops. Conventional breeding allows you to a certain level, But if you want to go beyond that, if you want to accelerate the pace of this genetic improvement of crops, we need to use all such technologies, and I think, yes, we have all those technologies available, and I would only say that they should be utilized fully to realize their full potential, in a very sustainable and responsible manner.
Great, thank you. Shivo, do we have questions from YouTube? I think you're on mute.
Yes, I put one question because Sir mentioned the cryopreservations of seed. So someone asked to it is viable or something. You can see the question, and the rest of the questions is covered by Sir already, or it may be covered by interview section.
So what is the question you're talking about? Cryopreservation of seeds?
Jacinta?
The cryopreservation question. Okay, is cryobank technology established? And what kind of plant or plant tissues have been successfully stored for this technique?
See, we are doing 3 different kinds of storage, actually, when you talk of cryopreservation. One is of the seed itself, wherein we conserve those seeds in the temperature ranging from -18 to -20 degrees Celsius in refrigerated storage conditions, providing electricity as a source of energy. And then another is we conserve them as in vitro cultures, in tissue culture, tubes, as in vitro cultures, which we cannot regenerate those plants, let's say, through, seed directly, for example, some of those fruit crops. And the third is we conserve them in liquid nitrogen. The temperature is as low as -196 degrees Celsius, So we are using these 3 different modes, and depending upon which particular crop, whether it is directly possible to regenerate, have a whole plant from seed or plantlet or otherwise some, part of a tissue, we conserve it accordingly using 3 different modes of conservation. And permafrost, in fact, is therefore a successful, Svalbard seed vault because it is maintaining naturally that kind of temperature which is required with minimal use of energy, near North Pole in Spilsberg, island near Norway.
Great, thank you.
No more question. Thank you, sir. Thank you, Jacinta. There are a lot of questions, I know, when you talk about the GM and GM technology, but I suggest everyone don't worry about the GM technology. As Sir mentioned, that there are lots of and extensive work has been done on that technology, and many articles are available. You should go for the data, go for the article, go for the analysis, then you think whether it is good, not, or your judgment based on that available data source. So thanks, your even I don't I follow your work a lot, but I somehow miss your chloroplast work. It's a great because sometimes and I feel the thrill when you saw that it is that time it's not available. When I work on jute, it's also happened to me. I have to isolate the housekeeping genes of jute because it's not available in such a way. So these are the little small steps, but it's very important to extend our field of research, field of that crops like wheat, maize, rice, whatever you've done. It's a wonderful as a lifetime work, and a lot of things we should learn from your work and your passion and the openness you have created towards the science. I ask Shoma, thanks. I request Shoma will do a short interview to know better of you, and many young students and scholars are listening interview sessions. So
they're around still.
Yes, they're around still, and so I asked Shoma, and video is available just after the webinar, so many people are coming around in their convenient time. So, Shoma, please do the interview.
Yes. So we are now moving on. Hello, sir. We are now moving on to the interview section of today's webinar. Today is our 20th webinar of the series, and we are so happy that you have joined us. It was a wonderful and so informative. Thank you for the presentation. So, sir, just to know what you think about, some things that I'm going to ask in the interview session. So, first of all, my first question to you is that um, you have done amazing work in the field of functional genomics and gene discovery, plant genetic engineering, plant physiology, plant genetic resources, and so many works. And we would love to know how you built your career. Could you please enlighten us about your journey so far as a plant scientist?
Yeah, well, I said before, that I had this passion of, taking science as a profession. And I must also say that after I did my graduation, I not that thought of becoming a scientist. Or even my parents, I would say that time parents also decide what their son should do.
Mm-hmm.
And maybe because of their influence or otherwise, I thought, our son will become a doctor. Or of course, I did appear in one or two, pre-medical tests,
Mm-hmm.
at the national level.
Mm-hmm.
I could not get selected. I was selected only for BDS, that is Bachelor of Dental Surgery, and I was not interested. That was my decision.
Mm-hmm.
Did not become a dental doctor. I wish I had become one to a lot of money now. But that time, I'm talking in 1972, 74, so I didn't want to take up dental surgery course.
Mm-hmm. Mm-hmm.
So after my BSc, then what I thought, again, without the influence of my parents, to become was an IAS officer.
All right.
And because I did not get admission in any of these medical colleges, and I didn't want to go become a dental doctor, and I did not get admission in masters. So what I thought I should do, I thought I should start preparation for my IAS, and we had a small room, on the first floor of our home, and that room I occupied completely and made a place for myself to study, because, you have to study very hard for IAS examination.
Mm-hmm. Yes.
So I started preparation badly for this IAS examination. And after a few months, I came to know that I got admission for a master's in Haryana Agriculture University in Plant Physiology Division, botany department.
Okay.
So I was also at the back of my mind that time thinking to have degrees, because I won't be only satisfied with having only a BSc degree with me, even if. So that attracted me to join master's degree in HIUSR. And initial 6 months, I was continuing both with, master's degree as well as IAS preparation. But after 6 months, I was totally spoiled by science, and that had attracted me a lot. And I said goodbye immediately to my preparation for IAS. And, one of my teachers that time, Dr. Krishnamurthy, said, you can be an excellent scientist, he thought. And then I thought, okay, I should pursue more of science, and it will give me an opportunity to earn more degrees. Mm-hmm. And I was fortunate to do my master's degree with Dr. O.P. Garg, one of the renowned scientists again, of the country, and who's again, of course, no more. So I did my master's there. And then I was selected as ARS for this, agricultural research examination in the same year after I finished my, master's degree. And then I joined Indian Council of Agricultural Research, and the rest is the history. After my, of course, this ARS selection, I was posted in Hyderabad for 1 year. And I had no experience of working in the field. And there the institute wanted me to only work in the field, and which I was not prepared to.
Okay.
No such experience. I was a pure science BSc graduate.
Mm-hmm.
And but then, I got a transfer to CPRI Shimla, Central Potato Research Institute Shimla.
Okay.
And but then that was good in a way, because my 1 year got wasted in Hyderabad, where I was placed initially, my selection. Mm-hmm. So that 1 year wastage gave me a big kick that, you should not waste your time when you accepted position as a scientist. So then I started working hard. From there I learned, and after I joined my PhD of 7 years of my stay in Shimla, that was from 77 to 1984, I was in CPRI Shimla. 84, I came down for my PhD to Indian Agricultural Research Institute with Professor S.K. Sinha. So from there again, I further learned the advantage of hard work and analytical thinking as a scientist,
Right.
and then, of course, I developed more and more interest in science and developed more of passion, and then started thinking big, that how do you now go to a lab outside India, to one of the best universities. And the Department of Biotechnology gave me an opportunity, to unfortunately, I was accepted by the professor in Harvard University, which was not easy again, and so that actually then prompted me to go further and do all the science, which I could little bit contribute so far.
Great. Do you think we should consider abiotic stress more seriously to ensure global food security during the times of climate change and global warming?
Yes, of course, this is one of the areas where we need to contribute more, and we have not been successfully, actually speaking, given enough yet, for some of these areas which remain rain-fed. We have, of course, contributed, I would say, significantly in terms of, hybrid vigor or increasing the yield per se, no doubt, under optimal conditions of irrigation with enough of fertilizers, etc. But then I think climate resilience, and particularly there's such traits related with climate resilience like tolerance to drought, salinity, high temperature, is something that we have not been able to achieve a lot, I must say, because either through conventional breeding or even through some of these advanced tools of GM or genome editing, because these traits are quite complex in nature. When you talk of breeders selecting for which trait, when we want to select for drought tolerance, or for high temperatures tolerance, this trait mostly is again the yield, advantage under those, conditions, but that's not a very good parameter, actually speaking, because those yield contributing traits, sometimes they differ depending upon under which environmental conditions, the crop is being selected for, is grown, because that environment keeps changing every year, So therefore, I think we need to, yes, make much more progress in this area. And now, fortunately, we have these facilities of phenomics, food phenotyping under controlled conditions. We have these tools of genomics, and we have all these tools, I said before, of developing genetically modified crops or genome edited crops. I think so in networking mode, we can try to tackle and address these issues in a big way. I'm sure we can achieve a greater success. And I'm sure already there are signs, while we have all attempted to develop some of these, genetically modified crops with 1 gene or 2 gene or 3 genes, for various abiotic stress tolerant traits, but still there's not that big advantage we have still achieved under, large tracts and field conditions in any of the country. So you're right. I think this is one of the areas we must, invest more in a network mode using all such tools and technologies around us and see that we succeed.
You have served as an officer on special duty, National Agricultural Higher Education Project, ICAR. Do you think higher education or education in general needs more focus on GM crops to encourage students towards GM crop research and to help develop scientific concepts about it?
Actually speaking, in our country, leave alone GM crop science at the moment, we need a greater emphasis and focus on higher education itself.
Oh, yes.
We need to strengthen our approaches to have bright young students coming to agriculture field. We must try to attract these talented youth, to agricultural sciences, and we must try to, modernize. And in fact, Division of Education of Indian Council of Agricultural Research is doing all such efforts in attracting, retaining the young talents, towards agricultural sciences. And we have 75 odd agricultural sciences in India, state agriculture universities in India. And this particular project you're talking about, it National Agricultural Higher Education Project, funded by World Bank and both Government of India. I was instrumental, as you mentioned, as OSD for only about 1 year. And that 1 year was quite significant for me because, with the efforts and guidance that time of Dr. Mahapatra again, who had just taken over as the General, of Indian Council of Agricultural Research, we were able to have this project of 1,100 crores of rupees, $165 million, and now, in fact, it was launched, I said, in July 2017, and about 3.5 years, it has made tremendous progress. And World Bank is quite satisfied, I was told, about the activities under this particular project. So when we talk of, education in general is important, we must try to lay much more emphasis in India because our economy depends to a quite large extent on agriculture, So we must try to have education, standards further raising high, excellence to bring into it, relevance, entrepreneurial skills as well, to have startups more in agriculture by some of these agricultural graduates coming out from these universities. I think there's a lot of future, bright future, I would say, for some of these agricultural graduates. And then comes, of course, their involvement in using the tools of science, technology, whether it is genetically modified crop science, you talk about it, or genome editing. Yes, of course, we need, many more scientists. But then more than that, we need, when we talk of these advanced technologies, I again urge a political will and commitment, to have this sustainable agriculture, which is good for the environment, in the coming environment, it's important. So education is extremely important, and we must play as teachers as much big role as possible to help these students and communicate, the good science to the society at large through these, state agriculture universities or educational institutions in the country.
Wonderful. Sir, this is a similar question. You're this positive person, and what motivates you to work for the betterment of agriculture?
Actually, what motivates me to work for agriculture is that we have just one life.
Yeah.
Okay. and our way of life is that you must make use of your life for the betterment of others. Okay. Of course, including yourself first, So having got all that knowledge, going through all those degrees I was talking to you about, if you want to put some kind of a use of all that which is worthy of, of contributing something, and advantage of becoming a scientist is what? Advantage is that you are having a fresh thinking always and a passion of thinking new and doing something new.
Yes.
That motivates you even, see, after you retire. I don't think a scientist will ever retire if you are really a scientist. Your thinking process will always continue, and you should have that passion. I would say youngsters have even a better prospect than what we had because of this technology around them, because of this internet, because of this closely knit up world, a global village, and we try to communicate, as much knowledge and disseminate it to those who need it more than us, to make their life better, in terms of providing a product or a technology or even communicating simply, so there's a lot that one can gain out of what you've learned over the time if you pass it on. I said before, sharing is caring, So I think we should all believe it more. And our Indian culture of life is always that we help each other more, we try to. So from that passion, I have that kind of a feeling, that if we can, really speaking, can help each other in a networking mode, and together if we make a progress, we all grow together, So that's the passion, which keeps me actually driving. And of course, I do learn from youngsters, from my students. I've learned a lot. I must say this, some of these technologies, for example, I have no idea about bioinformatics tools and kind of information technology, but then they are much ahead of you. And then you can combine, these biological tools, biological science with information technology, or with bioinformatics, or with artificial intelligence. We must have a concept clear that with our knowledge where we want to reach, so you must dream big. You must have something in mind new, but I'm sure resources are there, the intelligence is there, the networking, the institutions are there. Enabling environment, of course, need to be created, which is very important. Where we lack at times, I must tell you, our Indian culture, which is rather important for even doing better than the best.
Talking about India and progress, you have initiated many research projects on transgenic crops. Do you think India is going in the right direction in developing in this field?
Not really. We have already the bus. I must tell you this. Very frankly, I'm telling you, simply because I have witnessed in the last at least 20 years, we only have single BT cotton so far. I feel pity on myself and on those poor, I should not say smallholder farmers.
Yep.
They could have reaped the benefit, they could have harvested the benefit of many of these genetically modified improved crops. Now, what is keeping us back? I feel very bad about it. We are not certainly on the right path when you talk to me about the use of genetically modified technology or crops in India. Our Indian farmers, why should they not be giving those benefits? I can change my mobile every time, every day, a new version I can get, of any such technology. I can buy a new laptop whenever I feel like. But why our farmers are not able to buy those new seeds with all those genetically improved traits? For example, GM mustard. Why our farmers are not able to take the benefit of the GM mustard with hybrid vigor, using the same technology with which canola has been developed in Canada, and which is almost 100%, I believe, used by the farmers in Canada, that kind of technology and that GM canola, what we have in India. But our farmers are deprived of this technology, of these hybrid seeds. So I think much more, need to be done. And the role of scientists like you, youngsters, we should try to communicate it to the fullest possible through different ways of media, that we should embrace this technology, we should accept, and help our farmers grow this technology, of course, all through official means, and as much as possible, we should try to do it.
Sir, Bangladesh is growing BT, brinjal, aubergine, eggplant, whatever you call it. Can India and other Asian countries use their safety data?
Yes, very much. You know what? The whole world is a global village. If I find something safe, safety is tested by what? By laboratory tests. By making tests, doing tests on those material which we're consuming, which has been developed. If you do it in India, you do it in Bangladesh. And therefore, actually, I look forward to some kind of harmonization of all these regulatory, lines across the world. But unfortunately, it is not happening.
Yeah.
Even I would say for genome-edited crops, that harmonization is needed. If a country has found, we must have that element of trust, in science, So I don't know what more we need to do as scientists. There must be something that we are really lacking. We're not able to convey to the public at large, across the globe, that why science is not able to make as much progress with the genetic modification of crops, than other, walks in life. So I think there's something we need to worry about it. I do worry about it myself, I must tell you. And I wish there is a kind of environment created across the world that there is a lot of trust in science, and we develop these products for the welfare of not one person in one country, but all across. All countries are alike, metabolism point of view. So I think we need to develop such mechanism globally and promote, that it reaches to all those unreached, the last mile, or those poor, unnourished, children, of any corner in the world. They should all benefit of it, and sooner the better.
Yes. So what do you think? How many years does India need to commercialize GM food crops?
How many?
Years.
Oh, how many years do India need to
India
commercialize?
Yes, food crops, GM food crops.
Why you need, we're talking of years? I would say it should be released tomorrow.
No.
Yeah, I mean, like, I'm not, waiting for years. We have waited years.
No, we are waiting. So, I mean, it won't happen overnight.
It can happen. It's a question of only, I said, political will and commitment for the welfare of the society at large, for those farmers.
Right.
You
I
know,
mean, it should, yes.
and for the economy to grow, for making the environment clean. We're talking of organic agriculture. This could be an integral component of organic agriculture. You know, with the
Yes.
UT cotton somewhere in US, I believe they've been able to eradicate that pest as an integrated pest management technology, of which BT cotton is one of the components. So we can do wonders, you know.
Yes.
So therefore, I'm saying, sorry, not years, not even hours. We should
Do
do
it.
more.
Yeah.
If you're talking to me as a scientist,
Mhm.
as a citizen of India, as a concerned, responsible person, as a senior citizen also, if I may call myself.
Yes, I would eat GM crops, no problem.
Yes.
All right. So, do you consider CRISPR crops as GMOs?
Well, it depends, you know, which type of CRISPR mutation we are talking about it. If we are talking about SDN1, you know, which is the repairing of the double-stranded DNA break as a result of non-homologous end joining, which results into some kind of a insertion or a deletion of a variable length of any targeted gene to mutate, you know, their gene function. It should not be regulated at all. It should remain non-GMO. And moreover, such crops will be indistinguishable from those which have not been mutated, you know, using this technology. And we have been eating or developing varieties world over, thousands of different crops by random mutagenesis through chemical means or through radiation. So this is even a better way of mutation. I'm talking about this NHEJ, non-homologous end joining of a double-stranded break caused by, you know, this CRISPR-Cas9 technology.
Mhm.
So that should remain a non-GMO. Even other one, that is SDN2, wherein based on homology-directed repair, if you are only mutating certain known bases, okay, or you are adding certain, you know, kind of bases into it, into the existing, you know, to make existing gene mutate or add a function, you know, SDN2 category, that should also remain, you know, should be treated, I would propose and suggest, as many other scientists are doing it, many other countries have already done it, for example, US, as non-GMO.
Yeah.
The third category, where you replace the whole gene or you add altogether a foreign gene, you know, therein, yes, as a, you know, normal classical GM technology, it should be regulated. But there also the regulation should be minimal, because here we are targeting onto a particular predetermined location onto the genome, the integration even of a foreign gene, which was not possible or is not possible with our current use of genetic modifications, either through acrobacterium transformation or ballistic gun or otherwise. You know, because so far, whatever GM crops we have developed and consumed all over the world, you know, they get integrated at random onto the genome, you know. But here in this technology, you know, even if that SDN3 allows you to have a replacement of a whole gene or insertion of a foreign gene, but onto a targeted location onto the genome. So there, yes, it should be regulated, but again, minimal regulation, I would suggest, as compared to the current regulation we are watching, or, you know, through genetic modification methods.
Right. Do you think anti-GMO campaigns are damaging or misleading and are the main reason for delaying our country's commercialization process?
Actually, I would leave it to them to decide this. They are as intelligent human beings as we all are. And it's better if they comment on it. And if they feel they're doing right, and if it is helping, you know, in better world or better society, I'm sure they would have reasons more to tell us about it. But as a scientist, what I told you before, you know, that I would like to use these technologies in a very comprehensive and a responsible manner, and try to see that, yes, only the best products, the safe products, even at times safer than conventional bred products, reach to the market, to the consumers, to the society at large, which is our major focus of any advancement.
Mm-hmm. Yes, I have noticed something, you know, in many videos, you know, in social media, that GMOs are sometimes, they mix up. They think that GMOs are like pesticides. I mean, the GMO and pesticides are something, you know, similar things. Like, it's GMO free. Oh, it's good. So it's also just a label, you know, it's GM free or no GMO, non-GMO. Like it's a good thing. And
I got it, Soma, what you're trying to say. There, people call some of these crops as pesticide producing crops. You know, yeah, in a way, pesticide, for example, BT cotton. You know, what it produces, what is BT or that cry gene, what it is. It is from Bacillus thuringiensis, which is the source of this crystal protein.
yes.
You know? And this crystal protein has a role to play and cause havoc for that particular Lepidopteran insect or any other class of insect, depending upon which class of BT protein we're talking about here. So what we are expressing in a plant is not a pesticide.
Mm-hmm.
As I understand.
Yeah, yes.
We are expressing in a plant a protein, which in this case, you know, BT product is a cry gene, you know, and it doesn't create any kind of a harm to human beings. We eat thousands of proteins from plant sources, from animal sources, from other sources, right?
Right.
So similarly, this is one more protein. If I'm eating, let us say, you know, BT papaya, okay, which is not there, of course, or if I'm eating corn, for example, you know, or if I'm, you know, eating, you know, the seed oil of BT cotton plant, what I'm eating is the protein.
Right.
Right.
Which we can degrade in our stomach.
Right. And this protein is only harmful to a particular insect pest against which this has been developed. I mean, the technology has been verified, you know. Even that protein will not kill another class of insect against which it doesn't work. You know, there are specific receptors for specific, you know, proteins, you know, against which from a particular class of these insects. So we don't have that receptor to which this particular protein, cry protein, will bind and therefore create any kind of a problem, which has been tested, I believe, a number of times as a mechanism of action of these BT protein, you know, against insect pests. It only acts against a particular insect pest. So I think there should not be any such problem of, you know, GM plants being treated as pesticide producing plants or otherwise. Yes, they have inbuilt resistance against those pests in the form of, you know, this mechanism of action I described. You know, that's okay.
Yeah. Yeah, I hope people get it soon.
Yeah.
Okay.
I hope so.
Yeah.
Myself,
you have many strong international collaborations and responsibilities. How do you manage it?
we try to manage it with ease. You know, because first of all, you know, when you are networking, why only nationally, even nationally, and you can do that all with ease, provided you are very clear in your mind and thinking process, and you're confident, and you are trustworthy, and you're honest and sincere.
Right.
You can take any number of collaborations, networks forward with you happily, you know, and that's what we scientists believe in, you know, together, because no single scientist can really do whatever even I presented was at all, not, I would say, you know, contribution to a particular person as me. No, it was all because of the efforts of n number of scientists. When you talk of that big effort of wheat, scientists of National Bureau of Transgenic Resources, not only scientists, even those, you know, those lab attendants, you know, those laborers in the field, you know, they all contribute as much as we do, even more than us. You know, we have a defined role, you know, each one of us has been out there for.
Yes.
So it is a contribution of some of those students, you know, and I'm happy in a way they learn, you know, we develop these human resources, they become professors tomorrow. So it's a collective effort together always, you know, for anything, whatever we think. Thinking could be only yours, but when you implement it, it cannot be just yours alone. It has to be together in a network more. So therefore, these collaborations, you learn quite a lot, and we must believe in collaboration, you know, in a very honest and you know, and good collaboration always allows you to work better than doing alone. As scientists, I'm talking about.
Beautifully put. Sir, you are a reviewer for so many renowned journals. Can you give us some key takeaway messages to improve manuscripts?
Well, I think one has to have, first of all, a good writing skill. And at times, before you start even writing, as I normally do, we try to make a story in your mind. Okay. It should be very clear. So, and if you develop that kind of a habit of making a clear story in your own mind, putting all things together in the form of a story, you know, then you start sitting and writing down, and and of course, writing down in your own words, and as simple language as possible, no jargons to be used, okay, and sentences to be, kept small so that anybody can understand it easily. Not everybody can, understand English as we normally do. The medium of exchange of knowledge so far globally has been English, the part of the world. So not that everybody would intelligently enough understand it. So it's better that your message will go around to all those people who have, little less knowledge, for example, of that particular foreign language for them, it's a foreign language. So therefore try to, and of course, if somebody can assist you with regard to, editing it, from the English language point of view or otherwise, I think it should be fine. That's what I believe. And there should be clarity in your thoughts, actually, and you should try to see that, whether you are able to understand. Somebody else has to read through the whole paper. So whether they will understand it while they're reading, as if you are an outsider, yourself, and try to understand, do you get that message so clearly? Understand that first, again and again, and then, just not you write it, leave it as I normally do for about a week or 10 days, come back to it, so you will find yourself your own mistakes. And then, so therefore it takes time, to develop a good manuscript. So I think there are people, of course, I'm not an expert, I would say, in this particular area. My papers have been rejected, of course, many of them. And we have revised so many papers. So I'm sure there are better experts out there and there are better, I think, material available to go through this and to know more about it. But whatever I thought I knew, I just put before you.
Okay. According to you, which sectors of plant science research will prosper in the near future?
Which sector of plant science research? You know what? According to me, if we can win over two processes of plant sciences, and we can try to manipulate them at ease, I think that would be excellent. Though we understand those processes very well, not to the full extent possible that we can start manipulating it with ease at our will. One is photosynthesis, by which we get all that radiation, from the sun and, from the air, and we can, capture more and more of the carbon and utilize it by the plants, and, all. That is one. And as a result of that, we can increase biomass. It can go for various other applications of energy production like biofuel, other food security, and of course, taking care of the climate change as well. And another is nitrogen. Through biological nitrogen fixation, even if we can have that, in our cereal crops or those non-leguminous crops, if we to incorporate at our will, through any method available, actually speaking. Certainly conventional breeding is of course not going to work, which we know it scientifically, but what I'm saying, genome editing could be potentially very useful there, So if we can, according to me, understand fully well and try to not understand alone, but with an idea that we can try to manipulate it toward the advantage of plants' better growth with minimum resources of fertilizers or even taking care of some of those issues related with carbon and climate change. Because nitrogen, we put more and more fertilizers in the form of urea. And when you synthesize urea chemically, it also adds to the climate change problems. So, and of course, rice could be, let's say, for example, 100% non-aerobic or what do you call it as direct seeded rice.
Okay.
Direct seeded rice, aerobic, under aerobic conditions, without flood irrigation, can try to achieve those. Though there are already a lot of varieties, people develop, and I saw one trial in IARI where they're trying to have direct seeded rice. There are problems, but processes you talked about, I would suggest only photosynthesis and biological fixation to the fullest extent, if we can try to understand it from the way, from the angle of using it to our advantage.
Yes. Okay, sir, how can bioengine.com contribute to updating people about GM crops based on scientifically?
You're already doing excellent job.
We would love to have some suggestions from you.
This is number 28 series, seminar in the series. and I'm fortunate to be talking to you all. And I hope it will, slowly start making a bigger impact. And I'm very happy and congratulated they started a magazine online, which Jacinta mentioned. And I think this is excellent. You guys are doing very well.
Thank you, sir.
If any ideas come over, I would certainly be available anytime. We can discuss it over.
Yes.
And but sure, I think Bioingene has a long way to go. It's only a humble start, small beginning, and I'm sure you will further grow. And this is a very good effort, I must say this, particularly during this period, when you are restricted in traveling and utilization of all this technology. And thanks to those who discovered all this internet, Bill Gates. And,
yes, definitely. Thanks to them.
And the IT professionals, So thanks to them as well, actually speaking, to allow us to utilize our time, knowledge, sitting miles apart, thousands of miles apart throughout the globe. So I'm sure we have to find better ways as well, more means available and disseminate knowledge. It's important. You're right.
And as we are at the end of the interview session, we would love to hear from you some words of advice to the students and scholars who are new to the field of research.
Well, I think I have already said enough if they have taken note of it in between. And otherwise, the only thing I would like to tell them that they should try to have that confidence of doing anything new, and even if they have little knowledge gap or they have a problem in the language to convey, but they must always try to learn whatever the best from the surroundings and try to intelligently pick up any good idea and interact freely with any other scientist friend or student friend and develop a hypothesis and start working on it. Unfortunately, nowadays, frankly speaking, there's little problem of trusting each other, that this is my idea will be stolen by others.
Yes.
But somehow I'm of a different opinion altogether, that we should trust people as much as possible, even if, they're stealing your idea. Of course, one should not, but one at the most try to, work together. But what I'm saying is still, again, I think one should develop that habit of collaborating as much as possible, trusting people and trying to see that, yes, we can, take our own knowledge and use it and develop some new hypothesis, further down and develop on it. There are many examples, actually speaking, in our own country, whom they can emulate, not me. I mean, there are many other examples I'm saying, whom they can emulate and they can become examples themselves for others. I tell you, so we have all this ingenuity, that kind of a brain and try to, only thing is take it as a passion, that's something very important. If you want to do science, you have to have that passion. You can take it as a job. Though job is important, you have to have your own livelihood, you have to earn money, everything is important. But at the back of mind, you must also 24/7 have that passion of delivering something new, thinking new, and going out to the world that, hey, see, I have done this.
Yes.
And must come out generation by generation better than what the previous generation would have done.
Yes. So thank you. It was beautiful hearing from you, and you have given such fresh perspective to our viewers, our listeners. Thank you, sir. And I will like to go back to Shubho now, and let's see what he has to say.
Thank you. It was a pleasure for me for such a long session.
Yes, thank you, sir, for being so patient and bearing with us.
No, it's fine. I've enjoyed it so far. So good. Why not? It was
sir, it's already 1,125 people are watching.
Okay. I'm very pleased, and my regards and respects to all of them in any corner of the world they are. And well, if there is anything which I said and it was not, liked by them, or, you can always with me. I would request Mr. Majumdar to share my email with them and try to together make, this, planet a better place to live for not
yes, definitely,
but for all those animals, birds, and other organisms. We must try to see that we are more, compassionate and, here out as scientists to deliver.
Okay. Thank you,
To all
sir.
of them who's watching still.
Yes. So before you leave this session, I just start introduce to our executive editor, because I am very fortunate to have Dr. Sujan sir. He is from Houston Alpha Biotechnology Institute from USA, and I request him, can you help me
Actually,
to
Hudson Alpha.
Hudson Alpha. Okay. So I request him to, can you help me to jot down the Bioingene Ph.D. And I'm very fortunate that we are try to build, and he has a good publication in Nature and Biotech. So it will help us. And as you say, that writing should be a habit. And for Indian
Yes.
students, we need to be right from first day of MSc. I not say to for the PhD. It should be start from the BSc or MSc, as developed countries practices from so beginning. So it's another team. It's a PhD, and I'm fortunate to. So we are going to announce a little bit more about these things. So, sir, is a wonderful opportunity to have you. And always, as I am working on genetic modification crop, your words boost me. Every time when I
We
listen
must
your words,
continue
and
working. We must continue working in this area.
yes, sir. Yes, and yes, I'm sure a lot of people getting interested, and it give us to a freedom to when the gene pool is not giving us the right gene, we can get it from another options. So it's a opportunity we have, and we must go for it in a positive manner, positive way. Thanks, sir. It's a lot, as you say, it's a long session. Thanks, my
Thank
all
you.
team,
Thank you.
and we have some little bit announcement. So I request you. Thanks for joining. You can
Thank you.
stop. Thanks, sir.
Thank you, sir.
Thank you, sir. It was
Thank you
wonderful.
so much. Thank
Thanks.
you so much. Thank you, all of you. Thank you, Ms.
Okay.
Sita, Ms. Liberty, and all the viewers again.
Okay.
Thank you,
Thanks,
sir.
sir. Thanks.
Hi, thanks. Bye. Nice meeting you.
Thank
Nice
you.
meeting you, sir.
Yeah.
Have a good day.
Thank you. I'll catch up with you guys later. Bye.
Okay.
Bye.
Thank you, sir.
Bye.
Shoma, can you introduce Sujan sir, or introduce PhD a little bit, because we are getting so many submission. We are working on it, and I am thinking, I'm thought that it's Sujan sir, our as an executive editor, will be the best person to address, because many people have emailed me about the status, and we go back to it. And as I say, that quality is our priority. So it maybe take time, and we are working so hard on that. And because it is just starting, we have to finalize few things, other than it is called the back office work, or you can production work.
Yeah.
So we also need some time to that, but I'm very fortunate and very thankful to the participants, or to the authors who have submitted their manuscript. Some are outstanding manuscript, very good rating, good quantity wise, quality wise. So I'm very fortunate, and I ask everybody, please submit. It doesn't matter that which where and which volume you got, but I'm sure if it is work is good, and we are happy to publish it as soon as possible. But we try a format, we try to build a pattern, so it's very familiar to everyone, and we are working on our first issue. So I request Shoma, and then I request Sujan sir, you can elaborate little bit our scope of Bioingene PhD, and how someone can submit their manuscript. If possible, so you can share your screen to the bioingene.com website, or bioingene.com/PhD directly. Thank you. Thanks, Shoma.
So it was a wonderful session and it was our 28th webinar. Thank you all for making the webinar such a success. We plan to continue with all your support to make research available to more and more people. We are launching a journal. And we are so blessed to have Dr. Shujan Mamidi join the Bioingene team and mentor us, guide us, and take care of our beloved Bioingene platform. We welcome him as the executive editor of the journal. Dr. Shujan Mamidi is a senior scientist and computational biologist at Hudson Alpha Institute for Biotechnology at Huntsville, Alabama, USA. His primary research interest is genomics, and he works in various aspects like genome assembly, population, quantitative and functional genomics, among many others. He shares a vision similar to ours, and we are pleased to have him lead the journal section. Let's hear from him about our new endeavor. Sir.
Thank you so much for such a wonderful introduction. First of all, thank you for your continuous support for the Bioingene webinar series. As Soma mentioned, we started a Bioingene Plant Science Journal. It's an online, open access, international, and a peer-reviewed journal. The basic idea behind this journal is making research available to anyone or many more people, because most of the research papers are behind the paywalls. We want to break that barrier for some important research. And most important thing is it's free to submit. And we're anticipating the release of our first edition in a week or 10 days, hopefully this week. And for all the people who submitted, thank you for submitting it, and thank you for considering our journal for that. Since this is our first issue, we have been working hard to streamline our process, and we apologize for any delay in communicating with you or sending emails. As Soma and Shiva said, we're still organizing back office many things, the email templates or the journal formats. We're still in a process, so it takes us a bit of time to organize everything. And this is a completely voluntary organization, just run by just a handful of volunteers. So we are expecting all of us submit high quality articles that are edited and formatted as per the journal rules and that are not submitted elsewhere. One thing to make sure is no plagiarism. We don't want to run into issues either with the article or the journal. And on the top of it, we're looking for editors too, who are interested to spend an hour or two every month reviewing one or two articles. That helps you improve your knowledge, at the same time help promote our journal and science research as a whole. For now, we're only accepting article summaries that are published in major journals, reviews and mini reviews, perspectives, and certain concepts, important concepts that would be helpful for a broader audience. And we have a website built that has all the details. You could visit that at bioingene.com/psj. And if you have any specific queries, please send us to admin@bioingene.com. As I said, we might be a little bit late, but we'll definitely answer all your calls. Since this week is going to be a real rush for us, trying to organize everything for the first issue. Please bear with our delay, and please feel free to send us any questions and submit your articles. Thank you.
Thank you. And as Sir told, that we also looking for the editorial board, and we create a pattern that our editorials should have a H index greater than 5. So if you don't have a H index greater than 5, don't worry. We put you in our editor board, board of reviewers, because we also need reviewers. But obviously, we are looking for those editors, those reviewers, those have the PhD degree, those have the experience, those have the experience of international publications, and know the basic and few fundamental laws or rules of accepting a journal or articles. And many questions we got about the UGCQL list, but we are new. For the UGCQL list, we need some time. And definitely we are going towards to getting a impact factor journal. And we are communicating in such a way that your work will display it in every way. We have a group, it's called notification group. We are getting the registration on the group through the feedback form, through the registration form, through our email also, from our email, from our website. And we built already about 15K plus or 15,000 plus people in that group. So it's a opportunity to us. These are our first viewers of the journal. And it's not as we develop this journal, we customize a little bit in such a way that we can put the question answer, which is not available in normal journal. We also put the such things like the interview sessions, as you know that the experienced people, when we are interviewed, we got many positive, many motivationals, and many learnings from that interview. And we try to spread this interview in such a way, because not possible every time to view in the YouTube. So it's better if you have a PDF on that. So we are going in, these are the section, and we also put the sections for the career, like jobs, like postdoc position, PhD positions in that. So there are a lot of things, and that's why we got little bit time to establish at least for 1-2 issues. Then we can go. We may develop the protocol. Actually, in science or in our lab, we are in optimization or standardization procedures. When it's standardized, it's less time will require to get the result. So we are in the standardized positions, and we request everybody, please submit your write-up. It may be your review articles. It may be your summaries of a new published articles, or it may be mini review, opinions, anything. And keep it in mind, we are currently not getting the research articles. Original research article will start very soon, but as you know that research articles need more revision and more round of revision. So we are going included very soon in upcoming issues, and we let you know through the email and through our website bioingene.com. If you have any questions, you can go bioingene.com. You get everything, I think. If you not, then email me at admin@bioingene.com. Or if you have any particular questions for our editorial board, I can forward the email, or you can directly email to our editorial executive editorial. I will send you the link if you need to email him directly. So it's a new process, and we have some things like we collect the posters, because when someone write to going for the poster, then it's very difficult to start a poster. So we collecting the posters. We have few posters, and we giving the poster submission certificate upon getting the posters. But it display on our website. As you find in the footer, there are option of poster. You can go and see whether how it's look like. So thank you. Thanks Soma, thanks Jacinta, thanks Liberty, thanks Dr. Sujan sir, for it must be very late night to you guys, and thanks for coming. And as a team member and the team of Bioingene, we will try to promote plant science research. It's absolutely free of cost and open to all. Thank you. And those are expecting for the certificate, I already mentioned in the email that it will distribute it through our website. And go and visit our website. You can get everything from there. Thank you. So I request Shoma, you unmute, please.
I'll just stop the live stream and we can continue
Yes.
with the meeting if you want. We will just stop the live stream then.
Yes, you can stop the live stream.